high performance liquid chromatography. This is a technique for the separation of components of a mixture which are soluble in some selected liquids. For example, in this diagram you can see methanol is a liquid. Here you can see water is a liquid. Sometimes we may select a combination of both. Sometimes even we may use one more liquid. No problem. We can select even a combination of the liquid at a particular proportion of each. So the components we need to separate are soluble in these liquids to different degrees. Accordingly, we separate them. So high performance liquid chromatography. High performance liquid chromatography. This is used for the separation of non-volatile components, which means the components of the mixture cannot be evaporated. So in the liquid phase itself, they are separated. Here the mobile phase consists of one or more liquids. For example, water, acetonitrile, etc. Sometimes only one of the liquid is used, sometimes two or three are used, and whenever more than one are used, they are in a particular ratio. And these lipids are pumped at a higher speed using a pump. And the stationary phase is packed in a column. But the length of the column is less compared with the gas chromatography. Working of HPLC. The solvent is placed in a vessel and the solvent is pumped into the system using a pump at a predetermined rate. Then we have the injector. Into the system we are injecting the sample, a very small amount, for example 20 microliter of the sample is injected in the system. Then there is the column. The sample is being transported through the column by means of the solvent or mobile phases. And when they are coming out of the column, they are at different places because of the different affinity of different components towards the stationary phase. They are separated and they are detected and the data is transferred to a computer, while the spent sample is either released into a waste collecting vessel, or we can even collect that sample also. It is not like gas chromatography. In HPLC, if we want, we can collect the samples. Of course, the samples will be separated. Especially in preparative HPLC, we can separate the fragments or components, and we can collect them separately in different vials. In preparative HPLC, we can collect the fractions and not the fragments. Actually, there was an error. That's okay. The mobile phase of HPLC, which are liquids, are used in two ways. One is said to be isocratic, the other is said to be gradient. In isocratic method, solvent may be one or more, but always there is a fixed ratio throughout the process. For example, we say solvent A is 40% and solvent B is 60%. Then that 40:60 ratio is the same throughout the process. Then we call it isocratic. But sometimes during different time periods, we need to have different ratio of the solvents. Then we call it gradient type. And we have to select it in advance using a computer. We say in the first 2 minutes, 40% of sample A and 60% of sample B. Then in the next 2 minutes, 30% of sample A and 70% of sample B. Then in the next 1 minute, say 60% of sample A and 40% of sample B. Like that, if we need like that, we have to select it, and such type of work is called a gradient type. Examples of different solvents are acetonitrile, water, etc. and other solvents also are there. We select the solvent depending upon the components of the mixture we are going to study. The stationary phase of HPLC is filled in a column. The column may have a length of about 150 millimeter. 150 millimeter means 15 centimeter, the length of the scale in your pouch or in your instrument box. And the minimum length is often reported to be around 15 millimeters, which is very small. And internal diameter is 2 to 4 millimeter. Depends upon case to case. And the packing materials inside may have a particle size of about 5 micrometer. So the column is very short compared to that of GC. This is some data about the HPLC columns. Spherical silica particles of uniform diameter usually we are using. The diameter of the particle may be 2 to 10 micrometer, and these particles are very porous actually, and zirconia etc. may also be used. And the column is always packed in a stainless steel tube because high pressure is often used in that column. So the column should be able to withstand the high pressure being used. And these are the various data regarding this HPLC columns. This is the diagram of a column here. There are different types of detectors used in HPLC, and we select a detector depending upon the type of the molecules we are analyzing. Refractive index detector, UV visible fluorescence, conductivity, mass spectrometry, etc. are the different types of detectors being used commonly.